Hello, good morning. Today in Ciência Falada, our speaker is Carina Cunha. Carina is PhD in Health Science by the School of Medicine of the University of Minho. After she was visiting postdoctoral scientist at the Columbia University, and currently Carina is a junior researcher at the neuroscience research domain of ICBS. Carina, if you are ready, you can share the presentation and start.
Good afternoon to everybody. I would just like to start by thanking the organizing committee for the invitation to present my work here at Ciência Falada. And just to give you an overview and for you to better understand what's our purpose in doing research in the perception of natural rewards, I would just like for you to start imagining with me that maybe because it's lunchtime, many of you may not have had yet lunch and you wish to maybe go to one of your favorite restaurants to eat a very satisfying meal. Or maybe because we have been under very strict COVID-19 lockdown restrictions, you might be feeling a little bit lonely or a little bit sad, and you really wish to get together with your friends as soon as it's possible. Or maybe at your work, you are working towards closing a very important business deal, and you mainly want to achieve this because together with the deal, you'll also be gaining maybe a significant amount of money. But because many of these decisions and behaviors are part of our everyday lives, they are also fundamental for our overall well-being. But how does the brain allow us to reach our goals and actually obtain our desired rewards? So it's here that the brain reward circuit enters, and this is a highly complex set of brain structures that I have here represented in a very simplistic manner that comprises cortical and limbic structures to regulate actually our natural ability to feel motivated and to respond to specific desired rewards. And as you can imagine, because of the fundamental role in motivated behaviors in our everyday life behaviors, impairments or dysfunction in either one or several of these brain regions has actually been associated with development of neuropsychiatric disorders such as depression, substance use disorders, eating disorders, obsessive compulsive disorder, and others. And as you can also imagine, because of these, it's fundamental to understand how the brain reward circuit works in order for us to then be able to go back to disease and actually try to understand where the dysfunction occurs. And from this highly complex circuit, in the past years, many attention has been given to a particular set of dopaminergic projections that arise from the ventral tegmental area to the nucleus accumbens, because the activity within this circuit has actually been associated with our ability to perceive the existence of rewards in the environment, our ability to process information about these rewards, being these information either cues that will indicate the existence and availability of the rewards, but also past experience that we've had that will ultimately result in consumption of the rewards, which will be fundamental for our survival. And as you can see, the nucleus accumbens is able to integrate not only these dopaminergic signals, but also other cortical and limbic information and because of these, because being the center of this circuit, the nucleus accumbens is commonly named as the pleasure center of the brain. And in fact, this brain region in particular is composed by many cell subtypes. However, the biggest percentage, around 90 to 95% of nucleus accumbens neurons, are actually medium spiny neurons. And these medium spiny neurons are GABAergic in nature, and they project to effector brain regions, such as the ventral tegmental area and substantia nigra. And this to then be able to mediate motivated and goal-directed behaviors. One interesting aspect of these medium spiny neurons is that they can be divided either into those that express dopamine receptor D1 which are called D1 medium spiny neurons. And these neurons are able to project directly to effector brain regions such as substantia nigra and ventral tegmental area, thus being commonly called as the direct pathway. On the other hand, medium spiny neurons that express dopamine receptor D2 or D2 MSNs, they are able to mediate goal-directed behaviors. However, this occurs indirectly through intermediate brain regions, such as the ventral pallidum. And thus it's called, and together with D1 MSNs, which has a portion of neurons that also project indirectly to effector brain regions, compose the indirect pathway. Actually, and very interestingly, in the past and classically, D1 and D2 MSNs have been shown to have an opposing function to what regards motivation. And this is mainly because classical studies have shown that activity within D1 medium spiny neurons increases reward value and induces positive reinforcement. But on the other hand, activity within D2 medium spiny neurons has been associated with decreased reward value and negative reinforcement. However, and very importantly, in the past years and with the use of more up-to-date techniques, it has been shown that this dichotomous function is actually a little bit too simplistic to what regards the role of these neurons. And actually, some studies show that both D1 and D2 MSNs, they can regulate positive reinforcement. And to try to clarify this issue a little bit and to actually seek if D1 and D2 MSNs were could both play a positive reinforcement role and be associated with rewarded behaviors, we actually in our lab started by using behavioral tasks, which is called progressive ratio schedule of reinforcement. This was applied in mice and rats. And actually what this task allows us to do is that we present an animal with two levers, but only one, which is called here the active lever, and it's skewed by a light that is turned on above it. Only pressing in this lever will result in the delivery of a food reward. And very importantly in this task, what happens is that from trial 1 to the second to the third, the number of times that the animal has to press the lever will increase significantly. So the motivational status of the animal will be given here, because when the animal gives up pressing, this will result in what we call the break point. And this is actually a direct measure of motivation. And just a small note, I'll be referring to this behavioral task many times during the presentation. One interesting thing that we observed actually was animals that perform this progressive task, we could see a positive correlation in terms between the number of both D1 and D2 MSNs and the breakpoint of the animals, so the motivational status of the animals. And we did see a positive correlation with these two MSN populations. And so this data first showed us that actually activity of both D1 and D2 MSNs is necessary, at least for motivated behaviors. So having this information, this first data from our group and also previous data from other labs, started to make us question if actually there's a coordinated activity of D1 and D2 MSNs in the nucleus accumbens that might be crucial for appropriate motivated behaviors. So basically the aim of this project or this work is to understand what's the role of D1 and D2 MSNs in reward-related behaviors. So starting here with the first results that we got, we went to use a specific technique that it's called optogenetics. And what this technique allows us is to use these ion channels, which are exogenous to mammalian, in which we inject in our region of interest, in this case was nucleus accumbens, and allow us to specifically activate either D1 or D2 MSNs while animals are freely behaving, so are performing a behavioral task. And this technique as you can see by this representative graph, will induce a significant increase in the electrophysiological activity of the target neurons. And so we decided to apply this technique and perform a brief optical stimulation of either D1 or D2 MSNs when animals see the cue that predicts the active lever in the progressive ratio. And very interestingly, when we did this and when we compare the breakpoint or the motivational status of the animals in a session in which they received optical stimulation in comparison with a session where they did not receive optical stimulation, we see a significant increase in motivation when D1 MSNs are activated. But very interestingly, we also saw the same increase in motivational status when D2 MSNs were activated, thus showing that Q-paired activation of D1 MSNs and D2 MSNs results actually in increased motivation. Next, in order to further prove that actually D1 and D2 MSNs have a role in positive reinforcement, we decided to use a different behavioral task, which is called a conditioned place preference. And in this task, what we do is that we first habituate the animals to explore a box that is divided in three. So the animals are presented with three chambers, and they first are free to explore every chamber in the first pre-test session. In the second and third days of behavior, what happens is that we condition the animal. So we closed the animal in one box and we gave them this brief optical stimulation during the conditioning session in one day, and the next day we did the same, but for the other chamber with no optical stimulation. And very interestingly, again, and similar to the motivational effects that we saw, we see that optical stimulation of D1 MSNs will induce an increase in preference because the animals will spend more time in the post-test session in the stimulation-associated chamber compared with the no stimulation chamber. And again, we see the exact same tendency to increase preference for the stimulation chamber in D2 MSN-stimulated mice. Again, showing that brief activation of these neurons results in place preference. So the animals prefer the place where they receive stimulation. But then we decided to start to, in order to understand a little bit better what could be the mechanism behind this increased motivational effect that at least D2 MSNs had that hasn't been previously reported, we used optogenetics. Because, as I said before in the introduction part of this presentation, we know that D1 medium spiny neurons, which are GABAergic, they will project directly to effector brain regions, release GABA there, and maybe throughout this release have a positive reinforcement effect. And what we did was to do electrophysiological recordings in VTA, but also in ventral pallidum, because part of these neurons also projects there. And in fact, what we could see was an overall increase in dopaminergic activity in the ventral tegmental area, which is directly modulated through these D1 MSN projections there. And this has actually been described in literature. So increased dopaminergic activity in VTA has been actually associated with increased behavioral preference. However, to what regards D2 medium spiny neurons, they only project to VTA indirectly through the VP. So, but still we decided to record in both of these regions. And what we could see was that D2 MSN activity would induce a significant decrease in VP activity, which is consistent with the fact that they are GABAergic in nature, so they should inhibit their target regions. And actually, this caused an indirect effect over the VTA activity, showing a significant increase in dopaminergic activity here, again, going towards data that is shown in literature, from literature that shows that this will increase behavioral preference. However, and because as I said before, these data were very promising with brief optical stimulation inducing behavioral preference. However, we did go a little bit against what has been reported in literature. So to better understand what could be happening here, we decided to apply, instead of a brief optical stimulation protocol, to apply a very prolonged stimulation protocol in which from the 30 minutes of conditioning, the animals would actually have stimulation in 15 minutes of the time. And this is partially consistent with high frequency stimulation protocols that are used in other studies. And here, what was very interesting was that instead of having this increased preference, we actually saw a significant decrease in preference for the chamber with optical stimulation, showing that the animals would actually prefer to avoid this chamber to go more and to spend more time in the chamber with no optical stimulation. And because these data were a bit puzzling and actually the opposite that we had, we again turned to electrophysiology to see how this prolonged activation of D1 or D2 MSNs would change actually the activity within the ventral pallidum and VTA. And although we did see again the same effect, brief effect that we saw with brief optical stimulation, what happens is that after like 1 or 2 seconds, the stimulation starts, what happens is that there's a quasi-normal activity of VTA dopaminergic neurons when we stimulate D1 MSNs. On the other hand, and the other side of the story, when we stimulate D2 MSNs for a very prolonged period, actually in the ventral pallidum, there's a shift in activity that passes from inhibition to actually having a higher activity after the first few seconds of stimulation. And these data per se were a little bit puzzling, but then we thought that this could be, this electrophysiological effect and prolonged effect could be actually due maybe not to release of GABA in the target brain regions, but maybe other neurotransmitter. And here is where opioids came into our mind, because, and this is very important, both D1 and D2 medium spiny neurons, they are known to be able to co-release opioids. In the case of D1 MSNs, they are able to co-release dynorphin in the ventral tegmental area, and the release of dynorphin here will inhibit the activity of dopaminergic neurons. On the other hand, D2 medium spiny neurons, they are known to be able to co-release enkephalin, this opioid, if released in the ventral pallidum, will actually inhibit the activity of D2 medium spiny neuron terminals, thus usually inducing a significant increase in the overall activity of ventral pallidum. So what we thought that could be interesting to do was basically apply antagonists for these specific opioids in ventral tegmental area and in the case of D1 MSN stimulation, and the ventral pallidum in the case of D2 MSN stimulation. So we paired, in the conditioning day, we paired optogenetic stimulation, either brief or prolonged, with an antagonist for dynorphin in the ventral tegmental area, and in the VP, we paired brief or prolonged stimulation of D2 MSNs with an antagonist of enkephalin. And interestingly, one thing that you can see already is that these are the results for brief stimulation of D1 MSN and these for D2 MSN together with the antagonists. And what you can see is actually that when we apply these antagonists while we are performing a brief stimulation, there's no significant change in the results that we saw before. So there's still a significant increase in behavioral preference, either D1 or D2 with D1 or D2 MSN stimulation. However, when we apply D1 prolonged stimulation or D2 prolonged stimulation together with opioid antagonists, we lose the decrease in preference that we saw before, as you can see here with the stimulated non-treated animals in comparison with the stimulated treated animals, losing this kind of aversive effect. So these results actually allowed us to show that prolonged D1 and D2 stimulation most likely results also in opioid release. And this opioid release, it's what's causing this decrease in place preference. So at this point, and very interestingly, when looking again to literature, we could see that the data that we gather regarding the D1-MSN function is very concordant with most of the studies that we find in literature, showing a more pro-rewarding function of these neurons. However, still for D2-MSN role, there are still puzzling results and also recent results that are opposing to what we showed here. And we started to wonder if actually these puzzling and these different distinct results could maybe due to timing of activity of these neurons, because they might not be necessary to be active or inactive all the time, but this be associated with specific events during behavioral performance. Or even maybe there are different D2-MSN subpopulations that could actually have differential roles in motivated behaviors. So to try to tackle the first question, we turned again to the progressive ratio task. We validated again the brief optical stimulation associated with the active lever, showing that there's a significant increase in the breakpoint when animals receive optical stimulation paired with the cue, as you can see here. And this time we added a different group, a different behavioral group, that instead of allowing D2 optogenetic stimulation, will actually allow D2 optogenetic inhibition. And we actually see the exact opposite results as we see with optical stimulation. Then we decided to pair brief optical stimulation, but not with cue, but now when the animals actually receive the reward. And interestingly, when we did that, we show actually a significant decrease in breakpoint when compared with the control group, and the opposite was seen with D2 optogenetic inhibition, hinting that actually increased activity of D2-MSNs at Q increases motivation, but when it increases upon reward delivery, actually it will lead to a decreased motivational status in the animals. So next, we also decided to try to understand if all populations were responding similarly to behavioral events. What we decided to do was to use a distinct technique, which is called Photon Imaging. So here we are able to implant a lens in the nucleus accumbens of the animals, and we can actually see, using fluorescence markers, fluorescence calcium indicators, we can actually see change in neuronal activity while animals are performing behavior freely. And this work was done in collaboration with Rui Costa Lab at the Zuckerman Institute. And here what you can see is an animal performing the task. So it's pressing a lever and receiving here the reward in the center. You can see that the animal has the mini scope, which is what allows us to give us an image. And what you see here on the right is actually neurons firing. So these are calcium signals that you see while animals are performing behavior. And what this technique allows us then to do is actually to be able to align the activity, the neuronal activity, with behavioral performance. And when we did that, for example, for these D2-MSNs, in this case, what we could see is that there's a percentage of neurons that, for example, upon lever pressing will increase their activity. However, there's also another subpopulation of D2-MSNs that will decrease activity, and others that don't respond at all. And this was similar to what we saw actually when we align neuronal activity with reward delivery. We saw some neurons that would increase significantly their activity, others would decrease, and there's a significant amount of neurons that would not respond to this event. So this shows us actually that there are distinct D2-MSN populations that respond differently to the same behavioral event during a motivation task. So so far, what we could conclude with this work was that D1 and D2-MSNs, they play a significant role in motivation and reward behaviors, and this seems to happen in a time and cell-specific manners. So because of these, in the future, what we want is to understand how many populations do we actually have that are relevant for motivated behaviors. So we wish to segregate what are even within D1 and D2 expressing neurons, what's the anatomical distribution of the responsive neurons to the stimulus, and what's the genetic identification of these neurons? Because we clearly saw that genetic segregation using specifically or only D1 or D2 receptors expression, it's not sufficient to identify the function of these neurons. So lastly, I'd just like to acknowledge all the people that have been involved in this work, mainly Ana João Rodrigues and also Nuno Souza, who have been mentoring me for the past 10 years at the ICVS, and also the rest of the team here in our group and all the neuroscience research domain. And also to Rui Costa, who has been a precious collaborator of ours at the Zuckerman Institute, and also people involved in the work that I did there. Thank you.
Carina, thank you for your very interesting presentation. Can you please stop sharing the presentation so we can see each other better?
Yes, yes.
Okay, thank you. Now we have time to questions. It's open to discussion. Questions to Carina?
Maybe I can start with one question.
Okay, João, thank you.
Hi, Carina. So it's a really nice work. Of course, you're missing, your skins are a bit incomplete, I have to say, because you're missing these other cells, you know, the stars of the brain.
But not only astrocytes that I'm missing, there are other neurons there that I didn't speak about.
I was just wondering if they express D1, D2, or both, or if you have different populations of astrocytes. But this is just my question. But regarding your presentation, in the miniscope work that you showed, I noticed that in the first scheme that you had on top, the levels of activation would come down in one group of cells. They would go up in another group of cells. But the ones that are coming down, they already have a certain level of activation.
Yes.
Which is different in the
ones that go up.
Yeah. I'm not sure if you can show, sorry.
Yes, I can. Let me share again. So what we
do you think there is a meaning for this? Because if you can show. Yeah, exactly.
Yes, it's here, right? So here, I think you're speaking about these ones here.
Okay,
let me presentation. Okay,
yes,
so you see that these neurons that are here that decrease activity after lever pressing are actually already higher in baseline activity. While, for example, the ones that
levels, right?
Yeah,
to the levels of where the red were.
Yes.
You know? And this is different from what happens in the. So this happens during the lever press. Yes. And the next one is at the delivery of the reward. Right?
One thing that I want to call your attention to, just because, to be honest with the data, the y-axes are not the same because if I put it here, the exact axis as here, it looked like there was no change in activity, while when doing statistical analysis, there was. So, but regarding your issue on the baseline activity before, so I don't have a proper explanation for this, but there are some studies coming out showing that actually within medium spiny neurons in the nucleus accumbens, there are, although they are all very, they are quite inactive. So this is a very silent brain region, right? Because it usually only responds to stimuli, to external stimuli. However, what it has been reported is that there are actually segregated groups within medium spiny neurons that some of them seem to group in a higher activity, baseline activity in comparison with others. And it's not published, this data, but it seems to point that actually there are distinct populations, although looking like the same, so being D1 or D2 MSN expressing neurons, that they at baseline already present this significant difference in activity. So one guess of mine could be that these distinct populations then actually have a distinct role in behavior. For example, as you can see here, maybe if the populations that have higher activity at baseline are those that actually decrease upon lever pressing, and the ones that have the lower baseline activity are the ones that actually increase activity at lever pressing. Although so far I have no data to back this hypothesis.
Yes, we don't know if these are the same.
Yes, we don't. Sorry, can you repeat the question?
Or do we know? Can we follow the cell so we know if the
Yes,
ones
so
going up are always the one going up even at lever press and reward delivery?
Yes, we can. I didn't do that, but that's one of the advantages of using calcium imaging, for example, instead of fiber photometry, which give you calcium changes, right? But in imaging, you can actually image, you can see the cell, and you can follow the cell because you have a lens there. You can follow the cell throughout time and throughout the behavioral session. And if you're lucky, you can even follow it for many days. While in fiber photometry, you can only record fluorescence signals, so you couldn't do that. It's one of the goals of this work to follow actually the activity of these neurons in time, not only within the session, but also I recorded also the activity. So these are the data for the progressive ratio task. But I also recorded the activity of these neurons in the learning phases, because the animals have to learn to press the lever. So, and it would be interesting to actually follow them from the first day to the progressive ratio day and see if they have the same role in all of these behaviors.
Okay.
More questions? Okay, I don't think so. So I think we can finish now. Thank you, Carina.
Thank you.
And see you all in next week. Bye-bye.
Bye.